So our last talk, we have Blake Zhou, who is presenting research they did as an undergrad. Their talk is titled Brain-Wide Tracing of Inputs into the LP-ACC Reciprocal Circuit.
Thank you, Adam, for the introduction. Thank you, everyone, for being here. Today, I'll be talking about work I did in the last year in Marganka Sir's lab at MIT. And my work was on the topic of mapping inputs to the reciprocal connection between the LP and the ACC in mice using disynaptic rabies tracing. So I first want to start off by giving a brief overview of the two central brain areas in this work and why their relationship matters. So LP is short for the lateral posterior nucleus of the thalamus, and it is the rodent homologue of the pulvinar in primates. The LP is retinotopically organized, and it's highly involved in transthalamic pathways. It's mostly known for its role in regulating visual attention through dense connections to the superior colliculus and cortical areas, which is exemplified here in work from the Allen Institute, showing LP's reciprocal connections to various visual and frontal regions. For this project, we focused on the relationship between LP and the anterior cingulate cortex, or ACC, which you are all familiar with by now. It is involved in action selection and the allocation of attention. So since a main goal of our lab is understanding how the brain integrates sensory inputs to make decisions, we were really interested in finding out what information the ACC is getting from the LP and vice versa. Now, inputs to these two regions have been extensively quantified by other groups, but not with specificity to their reciprocal connections. So what we wanted to know is what brain regions are contributing to bottom-up processing from the LP to the ACC? And conversely, what brain regions influence ACC as it conveys top-down behavioral priorities via LP to other subcortical regions and beyond? To characterize the circuit, we used disynaptic rabies tracing to selectively target inputs to either the ACC to LP projection or the LP to ACC projection. And Lena did a really great description of the concept of the rabies virus with the deleted glycoprotein. The strategy basically involves injecting a retrograde AAV with CRE in a target region, but the CRE travels presynaptically, and we can take advantage of that to label a second node upstream of your target region with the rabies virus deleted glycoprotein construct, along with CRE-dependent helper AAVs, which enable the rabies to spread one synapse further into the rest of the brain. So here, just to recap, we have injected our AAV CRE into the LP, and rabies virus, along with helper viruses, into the ACC, with the result of brain-wide labeling of cells projecting specifically to the ACC and then to LP, which is why we have the term disynaptic rabies tracing. Conversely, we can also reverse the two nodes of injection, and we derived specific inputs to LP that then fed to the ACC. We were able to quantify these inputs by collecting images from across the whole brain, aligning each section systematically to the Allen reference atlas, and then annotating the location of labeled cells that we observe. So looking at the data, we found that the ACC to LP projection and the LP to ACC projection receive significantly different inputs. In broad strokes, ACC to LP receives most input from the cortex, whereas LP to ACC has less cortical input, but receives significant input from the midbrain. zooming in to the specific brain regions that contribute to the circuit, what is immediately clear is that LP and ACC projections are modulated by a diverse array of inputs. Starting with the ACC to LP side, the main contributors are frontal areas, such as the prelimbic area, PL for short, and infralimbic area, ILA. Associational areas also contribute a large proportion of inputs. So notably the retrosplenial area, which we heard about earlier, RSP for short. Looking at the LP to ACC side, the distribution is quite different. We see that while the cortical input is split between multiple sensory modalities, subcortical input is dominated by the superior colliculus. We can also look at this data by organizing it by function. So here we have cortical data divided in this way. Looking at it, we see that overarching differences between ACC to LP and LP to ACC in the cortex are more evident. We see the bias of the ACC to LP side towards these retrosplenial areas and prefrontal areas, as well as the broad spread of sensory inputs towards LP to ACC, which we'll get more into later. In the same vein, by summing up the regions that make up these cortical categories, we see what functions are important to the respective sides of the LP to ACC reciprocal circuit. I think this data also provides perspective to how LP is viewed, because LP is primarily thought of as a visual thalamus, but clearly there's so much more going on. And I also want to take a moment to point out that motor cortices also provide a significant input to both sides of this and should not be overlooked. So here we have primary motor cortex, and this is the secondary motor cortex. The visual motor associations of the circuit are emphasized by superior collicular input to LP-ACC. It is the biggest input to this projection. The literature around superior colliculus pulvinar projections largely regards retinal input and visual attention, which retinal cells project to the superficial layers of the superior colliculus. But looking in closer detail at the contributing superior colliculus subregions projecting to the LP-ACC circuit, we see that these inputs actually reside in the intermediate and deep layers and not in the superficial layer, which is interesting because while these deeper layers are still visually responsive, they are more strongly associated with motor and multisensory functions. These inputs from the superior colliculus to the LP and then onto the ACC also implicates the LP in a modulatory role in communication between the superior colliculus and the ACC. Because while the ACC projects directly to the superior colliculus, this is a one-way street. The reverse is not true. So LP could serve to provide a path for visual motor information to be filtered and returned from the superior colliculus back to the ACC. The LP and ACC also receive visual information from the cortex, but again, the inputs are asymmetric. Looking first at the top-down ACC to LP projection, the visual input is mostly from higher visual areas, namely the anteromedial visual area, vis-AM, and the posteromedial visual area, vis-PM. In contrast, inputs to the bottom-up LP to ACC projections are much more spread out between the various visual areas. And it's worth noting here that all anatomical regions were defined using the Allen Reference Atlas, and Vis-A actually overlaps with PPC a little bit. So the difference in visual input between these two directions is further highlighted by the broader context of other sensory modalities in these projections. Visual input dominates ACC to LP sensory inputs, and this dominance emphasizes the relative contribution of these higher visual areas. In contrast, in the LP to ACC direction, there are multiple modalities contributing significant input, which kind of overshadows the visual input, because you can see that primary somatary sensory input is much higher than primary visual input. And the potential importance of these non-visual modalities has actually been shown by work from other labs as well. There's recent work showing that neurons in the LP respond to whisking, which shows the somatosensory effects. And other work has shown that the LP can moderate auditory tuning via reciprocal input to the auditory cortex. So to summarize, today I showed you an anatomical mapping of the specific areas which influence the communication between the LP and the ACC. The ACC to LP projection is characterized by associational, frontal, and visual inputs, while the LP to ACC projection receives a broad multimodal array of sensory inputs, as well as superior colliculus and inhibitory inputs from the midbrain. Taken together, these data highlight the LP as a switchboard of sorts for thalamical cortical communication across many areas, and it provides a roadmap for understanding how the LP interacts with frontal areas to modulate the selection and allocation of attention. I'd like to thank everyone in the SIR lab for support in my work over the last year, particularly my PI and mentors who provided help and guidance throughout this project, the DCM staff for animal care, and finally Ian Wickersham's lab for providing virus. And thank you to all of you for listening. I'll take any questions now.
All right, great. Thank you, Blake. That was a great talk. Very impressive amount of work. We do have time for questions. So if people have questions, post them in the chat. I'll lead off first. It's amazing how diverse the connections are within this circuit. So you have really it seems as though it spans most of cortex. Is there anywhere that's relatively silent between these two regions? So is there kind of a space where you might say these areas don't get information from?
Well, like you said, there was a remarkable variety of inputs. I definitely received a on-the-go education on tons of brain regions I'd never heard about. I think the hippocampus is very sparsely labeled, which I think is expected. Honestly, I think we were also expecting more primary sensory input, whereas we see much more secondary and higher order input.
Yeah, that's really interesting.
I have a question. So you basically looked at the specific neurons that are, either receiving inputs from the ACC or sending out projections to the ACC. So what's the proportion of these neurons compared to the total number that is present in the LP and vice versa? Like, what proportion of LP neurons are the ones that project to ACC? What percentage of ACC neurons are the ones that project to LP?
So we can actually go back and look at LP and ACC in this data. So here we have LP to ACC to LP. Is this kind of what you're getting at? The proportion of inputs that you see? Or
the question was most not in terms of inputs, but because you kind of use this special strategy, right, for looking at the inputs that, if I understood it correctly, that you looked at the inputs that LP LP neurons that project to ACC, what their total number of inputs are, right? Like that specific, those specific neurons. And so I was just wondering, what is the total proportion of those neurons compared to the rest of the LP population?
I would say it's pretty sparse. We were targeting medial LP specifically, and just because of the methodology that you have to have the Cree virus travel, and it depends on all the viruses being in the same place at the same time. the specificity is very high, so I think it is relatively sparse, especially in the LP. I think the rabies virus was actually designed for cortical labeling rather than subcortical labeling. So definitely, yeah, more sparse in the LP compared to the ACC. But given that, there's a remarkable number of projections that resulted from that relatively sparse population, which speaks to how well connected LP neurons are.
Great. Well, that was a fantastic talk. Actually, 3 fantastic talks. So I want to thank our